Mhm. Okay, can everybody see my screen? Okay, so good afternoon, everybody. My name is Ruth Meletz. I am a third-year environmental science major. My faculty mentor is Dr. Ying Huan Lin, and today I'll be presenting the temperature dependence of emission product distribution from vaping of vitamin E acetate. So in this project, we wanted to look into vitamin E acetate vaping due to the 2019 EVALI cases, which stands for e-cigarette or vaping use associated lung injury. Patients who had EVALI suffered from inflammation and injury like infection and also tended to be users of illicit THC products. In the radiograph to the left, this depicts the lungs of an EVALI patient from the thorax and O'Carroll study. And I've highlighted areas in yellow that show signs of ground glass pulmonary opacification, a common indicator of injury in the lungs. According to the CDC, there were 2,807 confirmed cases and 68 deaths by 2020. 33% of patients diagnosed with EVALI admitted to using only THC vaping products, and 82% admitted to using THC at some point. Additionally, vitamin E acetate was identified in many THC-containing product samples tested by the FDA and other state laboratories, further supporting the role that vitamin E acetate played during this outbreak. These products were identified to be illegitimate, meaning that they were from unofficial sources such as friends, family, in-person, or even online dealers. In a study by Blount, researchers have also looked at the bronchoalveolar lavage fluid in EVALI patients versus that of a healthy group. They found that vitamin E acetate was in 94% of EVALI patients. However, vitamin E acetate was not found in the healthy group samples. Another point to note is that not all patients suffered the same level of severity as other patients. And this is because there are many factors that go into vaping. Vaping itself is very different from smoking because in this case, you're inhaling vapor, which is a product of vaporization, rather than inhaling cigarette smoke from combustion. And how this vaporization occurs in vape pens brings into account many external factors that may affect what emissions people are exposing themselves to. In a study by Sun, they saw that emissions in vape aerosol differ within brands of e-liquid, even if they are the same flavor, and found that these differences were dependent on specific aspects of the pen's construction. Some of these factors include the pen's power output, the airflow that the user uses, the diameter that the air flows through, the coil resistance, and even the stainless steel body of the vape pen. However, in this study, we focused on the effect of temperature. As mentioned in the previous slide, the power output can affect emissions because the power that the battery sends to the coil can affect the temperatures that the user will vape at. And the operating temperature of the vape device is one of the many parameters that a user can alter during a vaping session. Because of the power output dial in the vape pen, and what I mean by this is that at the bottom of most THC vape pens, there's a dial that lets you choose what voltage setting you'd want to vape at. And so this freedom in voltage choice can potentially affect the thermal degradation of e-liquids. And existing literature has already started to identify many volatile organic compounds such as ketenes, quinones, and terpenes in vaping aerosol. Volatile organic compounds are, which are known as compounds that have a high vapor pressure and low water solubility, have been seen to cause oxidative stress in human airway epithelial lung cells. And if you don't know what oxidative stress is, It is defined as a disturbance in the balance between the production of reactive oxygen species and antioxidant defenses. Reactive oxygen species are oxygen ions and free radicals that are highly reactive because they have an uneven amount of electrons. And our body usually produces antioxidants, which can neutralize these free radicals by donating electrons. But if there's an imbalance and there's a high presence of reactive oxygen species, this can cause cell and tissue damage, chronic inflammation, or even play a role or contribute to the development of EVALI. And so our main research question is, does variable temperature play a role in product distribution? To determine the temperature dependence of vitamin E acetate, we had to determine the peak temperature that the pen produces at each of the voltage settings available to use. We did 3 triplicate trials for the 4 voltage settings available on the pen, and these were 3.3, 3.8, 4.3, and 4.8 volts. And we did this using a data logger, a timer to help with activation cycles, and a spinner to vape pen, which was the model of pen that we used. And we had 3 thermocouple probes that were hooked up to the data logger, which would record the temperatures each second. So the first thermocouple probe, which is depicted by this yellow arrow here, was allowed to suspend in the air so that it could record air temperature as a control. Then we had 2 other thermocouples seen here in this graphic. The second was allowed to come into contact with the oil and record oil temperature. And then the third was inserted into the airflow tube so that it could come into contact with the coil head and record coil temperatures. And this was the temperatures that we focused on for the other experiments that I'll go into later in this presentation. And from our data, we then determined that the peak temperatures that the pen produces at each of the voltage settings, we saw that as voltage increased, coil temperature increased as well. And then we did an e-cigarette collection in order to capture the aerosol emitted from the pen and be able to determine what was being produced in the aerosols at all of the voltage settings. So how we did this is we had a cold trap that was inserted into a negative 40 degrees Celsius chiller. And then we attached tubing to the vape pen and also to a diaphragm pump, which whenever we would activate the pen, the diaphragm pump would pull the aerosol through and allow it to condense in the cold trap. Then we would do solvent extraction and then collect those samples and run them through a GC EIMS. And so back in the introduction, I mentioned that there are many different aspects that can affect vape aerosol, and recent studies have hypothesized that metals may act as a catalyst to enhance thermal degradation of vitamin E acetate. And because of this reason, we wanted to conduct pure pyrolysis of vitamin E acetate using a tube furnace to consider this possibility. And so what I mean by pure pyrolysis is we wanted to look at the thermal degradation of vitamin E acetate in the absence of any outside environmental factors, such as the oxygen that's usually present in the air when you vape, or any other gases present in the environment, any particles, or even like in the absence of the metal that's usually found in vape pens when you vape. So we just wanted to look at the thermal breakdown of vitamin E acetate in the absence of all of these external factors. And how we did this is we put vitamin E acetate into a little crucible and slid it into the tube furnace. The tube furnace would then ramp up to the peak temperatures that the coil peak temperatures that we determined. And then in a similar fashion, the aerosol or the emissions would get trapped in the cold trap, and we would then use solvent extraction to analyze our samples. So these were the chromatograms that we got from our emission samples when we ran them through the GC EIMS. And right off the bat, we see that there is a lot of degradation happening in the vape-induced samples, the normal vape pen like trials. And we saw that there is extensive degradation throughout the retention times and at all of the temperatures, even the lower ones. We also see that abundance does increase as well as temperature increased, But then when we look at the pure pyrolysis, so the tube furnace emissions, we see virtually no degradation up until the higher temperatures. And this is significant once again, because this may indicate the effects of a potential metal catalyst. But obviously, we see that there has to be something else that is causing such extensive degradation in, normal vaping emissions, but nothing in just like pure thermal degradation of vitamin E acetate. So from these chromatograms, we were able to use a NIST database and chemical analysis software to identify all of the different compounds found in the samples collected. And so this heat map shows some of the most frequently and consistently seen compounds, and visually shows the intensity of concentration each compound is found at throughout the voltage settings, depicted here by their peak temperatures. And so basically the main point of this heat map is to show how degradation of products was more extensive as temperature increased. And it's a bit hard to see in all of the compounds, but specifically in duroquinone, which I've highlighted here in red, we see that as temperature increased, there is more concentration being found. I also want to point out the fact that we were able to identify duroquinone at such low temperatures, some of the lowest in current literature. And as I previously mentioned in the introduction, duroquinone is a compound that has been seen to cause oxidative stress, and seeing it detected at such low temperatures showed the extent of risk that vitamin E acetate could have had on users. So in conclusion, we see that temperature does play a major role in the product distribution of vitamin E acetate, and we see that increase in voltage leads to an increase in temperature, and that with an increase in temperature, we see a more extensive degradation. But we also see that at low temperatures, there can still be exposure to harmful compounds such as duroquinone, overall showcasing the fact that variable temperature plays a role in exposure and the amount of degradation of vitamin E acetate. And so some of our follow-up studies to investigate some of these results further include examining the role of oxygen in the formation of thermal oxidation products in vitamin E acetate vaping, and also characterizing the extent of thermal degradation with metals that usually make up vape cartridges. And so I would like to acknowledge the NIH and the Mark U Star program for helping fund this research, and also the Lin Lab for being such a supportive environment, my faculty mentor, Dr. Ying Huan Lin, and my graduate student mentor, Alexa. They've also both been super supportive and have been great mentors to me throughout the entirety of this project. And so, thank you so much for listening. And if you have any questions, feel free to put them in the chat or unmute yourself. And yeah, thank you very much.
Thank you very much, Ruth. That was a really great presentation.